wonderful sight this is to have everybody here tonight. Thank you for coming on this rainy week that we're having. I can assure you that it was going to be worth your while to come to this tonight. I want to thank everyone for putting this together. The Komen group had put this whole thing together for us today. We're very thankful to them. They're so organized. I mean, look at the pink centerpieces and everything. I forgot my pink, but I have my Rutgers red on, so sorry. So in light of what's going on today, I have the distinct pleasure of introducing our wonderful speaker today, and I think you're going to love this talk. It's informative, it's cutting edge, it's what we're all looking for. Precision medicine, as you know, is taking on a new world, and it's taking on the new frontier for us, and certainly something that we're all exceedingly interested in and can't wait until it's all frontline for us. But I've had a few minutes to chat with Dr. Park, and he's just a lovely person, and I've seen his talk, and it's excellent. So a few things about Dr. Park that I want to let you know. He is a Midwesterner, so don't hold that against him. He's originally from Michigan and then went to University of Chicago. He had the good sense then to come east and went to University of Pennsylvania, and that is MD, PhD from there. And then from there, he ended up doing work at Johns Hopkins, and a little place down in Baltimore that we've heard of, another cancer center. So then he was there for a while, and he was after he did his postdoc work, and he became a medical oncologist there and served there for a number of years. Then he had the distinct pleasure to move further south to one of my favorite places in the world, Nashville. And he's at Vanderbilt University now, is currently in the Donna S. Hall Chair in Breast Cancer, the professor of medicine. He's a medical oncologist. He's also the associate director for breast and basic and transitional medicine. And he's the director of precision oncology there, which of course is mostly where our talk is about. He pioneered the use of liquid biopsies, which is one of the things that we're waiting for to move forward in our lives. Certainly as I'm Michele Blackwood, by the way, as a breast cancer surgeon. Some of these things are cutting edge for us as well, surgeons and medical oncologists. But a little bit about Dr. Park that you don't know, that I was able to get a little bit of the background information. More important than his MD, PhD, and all his incredible training is that he's an identical twin. Yes, and his brother named John, and he'll tell you probably a little bit about John. John's just mildly successful and retired with no mortgages multiple years ago out in the Bay Area. And there's two things he likes. One is he likes good beer and bad jokes. So if you have some bad jokes, I know we don't have any good beer here. So I'm sure, or if Dr. Alexander was around, maybe he could bring his beer that he creates. But without further ado, I'd like to introduce Dr. Park, and please give him a warm welcome.
Thanks, Michele, and thanks everyone for inviting me. This is the second time I've been at CINJ and Rutgers Cancer. The first time was well over a decade ago, and there's just been tremendous change and growth. I want to give a shout out because my former graduate student, Kelly Kuiker-Snowman, is here. She's a postdoc here now at CINJ, working all of the guys lab, four floors above, I think he said. And Kelly knows that I have this tradition at our holiday party that you have to tell a short, clean joke before we go to the gift table and do this kind of ridiculous gift giving ceremony. So I'm going to tell my short, clean, bad joke, since Michele already told you. All right, which one of these four things is not like the other three? I know you know this one, so don't give away the punchline. Yeah, I recycle these jokes. It's a lobster, a crayfish, a scrod, or I'm going to say a Korean man run over by a steamroller. And it's the scrod because it's a fish, and the other three are crustaceans. There's a backstory behind this, how I won a local Philadelphia radio contest for Worst Joke Wednesday. And I got a first. Wow. I need several more wines to tell that story. I don't even think I turned on my.
Sorry, that's your joke.
I can tell them, yeah.
I don't think you might not be allowed. No, I think you're fine.
Okay.
Yeah.
can also do this in line. Okay. Picture in your mind. My first slide, or next slide, will be disclosures, of which there are many. Probably many of you know now we're over disclosing these days, and I don't have all of these disclosures currently. They go back 5 years to present. I'm going to rifle through that once this is done. Anyone have another joke? Please just eat amongst yourselves. Okay. All right. Not working out. Oh, all right. Okay. Awesome. As I said, these are all not present past 5 years. Depends if you're from ASCO or ACR, et cetera. All right. I'm not going to belabor this slide, but I think many of us know these statistics. At least 250,000 women are diagnosed in the United States each year. The majority of these women will actually be cured, but 40,000 patients are dying every year, greater than 40,000. So this is a huge problem in our country. It still remains a problem. A lot of the work that I'm doing right now in the lab and in the clinical arena with cell-free DNA, I'm not actually going to talk about that because I've given that talk many times, and I think many people can look that up. And I want to talk about some new stuff rather than early stage disease, where we've been focusing our efforts on liquid biopsies, but metastatic, and actually thinking about new ways to target specific genetic alterations in breast cancer. All right, so the problem that we have for breast cancer in general is that we don't have one mutation that comprises 80 to 90% of all breast cancers. This was the case for colorectal cancer, where we know APC is a tumor suppressor gene, and probably 70 to 85% of those patients, of colorectal cancer patients, have some sort of mutation in that one gene. And the majority have it in the second gene. They're mutually exclusive because they're right next to each other in what we call a signaling cascade. But with breast cancer, we now know that it's a very, very heterogeneous group of diseases. And so with the exception of the P53 gene in triple negative breast cancer, there really is no one gene that is mutated in the vast majority of breast cancers. I think as a field, we have missed the boat in terms of our ability to target this. We're catching up. But our lung cancer colleagues have really led the field, where they've said, you know what, even if this is only mutated or altered in 1% of lung cancers, if we develop really good drugs, maybe we can make a difference. And 1% is still a lot of patients around the world. When I was writing grants as a junior faculty, if I tried to propose a gene that was altered or mutated in 5% of breast cancers and proposed to study it, probably would have just got kicked out of study section, because that was the mentality back then. And I think, again, we've matured in our thinking or evolved, that we know the heterogeneity between individuals, even if you have triple negative breast cancer or ER positive breast cancer, is very, very different. So with that in mind, I want to bring to your attention this case report, which I also probably warn you if you haven't started eating. The next slide is kind of gruesome and graphic. This is a case report of a very rare type of breast cancer called juvenile secretory breast carcinoma. It's usually cured with surgery alone. This was a case where it was misdiagnosed, had a number of local recurrences, and then also ended up becoming widely metastatic. It is not chemo or radiation sensitive, but this is really a testament to, again, science leading to translation and eventual therapies. So one of the things that these rare breast cancers are known for is having this particular alteration that we call translocation of ETB6 and NTRK3. So this particular patient's tumor was sent to MSK, where we're doing a trial of a particular drug, and they're staining for this particular protein of NTRK3, and then next-gen sequencing shows that one gene is inappropriately now next to another gene. That causes activation of the NTRK3, leading to the pathogenesis of this particular breast cancer. All right, so this was a 14-year-old girl at the time. This is her chin. You can see this is her third or fourth local recurrence. And she gets on this study of now FDA-approved therapy called larotrectinib, or LOXO 101. And in my earlier disclosures, just so you know, I sat on the scientific advisory board for LOXO Oncology, who made this drug and sponsored this trial. But this is a pan-TRK inhibitor, and you can see within 60 or 54 days in this example, it's completely gone. This is her CAT scan. For those of you not familiar, we're looking from the feet up. So this is the patient's right side, and this is the left, and that's that tumor mass. And within 2 months, it's completely gone. And here we're looking at lung windows where you can see all these brown dots. Those are metastases, and you can see they're pretty much completely resolved after 60 days of therapy. These are blood vessels that we're seeing. So this was really incredible. Has almost no side effects. This little girl is actually just popping, she's not a little girl, this is 2016. She's popping a pill every day and doing okay. And I think what was mostly the most amazing part of this story is how we actually found this patient. So this patient was actually from Bangladesh, from a small little farm. And I'm part of this monthly global virtual breast tumor board that is led out of Massachusetts General Hospital by my colleague Paul Goss. We actually meet every month. We just met this morning. I could only stay on for the first 30 minutes because I had to catch the plane here. And then I also happen to be on the LOXO Oncology Scientific Advisory Board. So I like to tell people I'm not really religious, but this story makes me believe in divine intervention because there was, I'm the only geeky physician scientist on the board. And so nobody had really ever heard of this. And I also remembered that there was a paper in Cancer Cell 15 years before showing these ETB6 translocations, and I happened to sit on the scientific advisory board and knew that we were looking for these patients for the company list. So she's back home popping a pill, doing great. And that really spoke to me of how much we could do to help patients today. If we could just reach them, get them the proper testing, and actually get the drugs to them. And so that's been a big part of my mission going to Vanderbilt. So with that in mind, what else can we look for? So this is work that I'm going to describe to you that involves many fellows who I always like to say spent the best years of their lives in my lab, slaving away. But it's been a fascinating story. So this is Brian Dalton, who's now actually on the faculty at Johns Hopkins, the junior faculty in the malignancies group. Brian, knowing that he wanted to study leukemias, wanted to focus in on genes that were mostly mutated in hematologic malignancies like myelodysplastic syndromes, acute myelogenous leukemia, et cetera, but also still had some low but real frequency of mutations in other solid tumors like breast, pancreas, lung, et cetera. And so SF3B1 is a gene that encodes for something called a spliceosome, and I'll go into that a little bit later in detail what that means. But one of the things in cancer we try to look for is what we call hotspot mutations, or regions in a gene or protein that are frequently mutated in cancer. That tells us that it's probably very important since this is a recurrent type of mutation. That doesn't happen by chance. It means that somehow the cells are selecting for a growth advantage if they have this particular mutation. And in this case, K700E, all that means is that this particular amino acid of this protein is mutated. And it goes from a K, which is a one-letter amino acid code, to an E. That is by far, at least in many cancers, the most frequent mutation. But most of them, the other mutations lie in this particular region or domain of the protein we call the heat domain. So what does this do? Normally, our genes, our DNA in every cell is a blueprint, but that DNA gets changed to RNA, and that RNA gets changed into protein. And protein are really the business end of what our cells are made of. In order to do that, there's a very complicated and intricate mechanism to change DNA into RNA, and we get rid of these sequences that we call introns. How does the cell do that? It does this by a spliceosome complex. And so SF3B1 is a gene that encodes the protein SF3B1 into this complex. And it will recognize certain sequences of DNA and then, or I should say RNA, and then splice them out so that you get a mature protein. It turns out, if you have a mutant SF3B1 or a mutant spliceosome, you don't excise that RNA correctly. And what can happen is this extra sequence here in pink ends up becoming part of the protein, or it can get degraded depending on the nature of where the sequencing, the splicing occurs, the misaberrant splicing. I'm not going to go into this part because it's not really relevant, but that's the kind of key. All of our cells normally DNA, RNA, protein. If you don't have a normal spliceosome complex, you're going to make abnormal RNA and you're going to make abnormal protein. So our lab for decades has literally been involved with taking cells, usually normal human breast epithelial cells, and then introducing mutations precisely at where the gene is, so we can recapitulate cancer mutations. So we don't do this by overexpressing a gene because that's usually not physiologic. It has some caveats. We can now genetically modify one cell so that we change just a single base pair of DNA to make it look just like a cancer DNA. Now, cancer is really the accumulation of multiple DNA mistakes. So what we've tried to do is model that. We introduce one mutation, and then the same cell, we introduce another one, and so forth. But we have the advantage that if we do this stepwise, one mutation at a time, we can compare it against the normal cell. Because those two cells are what we call isogenic. They're only different by one single DNA base pair change, just like what happens in cancer. Once we create these paired cell lines through various techniques now that have evolved, we can study them pairwise. We can look for differences in protein and RNA, differences in drug sensitivity and killing. It gives us a lot of tools and reagents to really study the biology of a given cancer mutation. And so Brian actually took this on in the lab, and he took 2 non-cancerous human immortalized breast epithelial cells, and he introduced these mutations. And so these are independent derivations. It's different cell lines, so we call them K700E1 and 2, 3 and 4. This is a separate cell line where he's done the same thing. He also introduced, just for as a control, where he did the exact same thing, but he just made what we call a silent mutation. So he genetically modified it, but it doesn't change the protein sequence at all. And that's why it's R702 for R. So there's no difference in the protein sequence because of the nature of how proteins are made from RNA. TWIT is not a play on words, it's targeted wild type. And so that is also a control cell line where Brian just introduced the same exact DNA base for the same exact. So these are all things that we have to do to make sure that our results are really reproducible and not an artifact from doing this gene editing, as we call it. So one of the things that Brian first did is once he created these cell lines is just to simply ask, does it in fact change the RNA? And so you can do this by something called reverse transcriptase PCR, where you can just look for the presence of new bands on a gel. So we can actually take these arrow, what we call primer sequences, and then amplify RNA and DNA between these 2 arrows. And if you actually have aberrant RNA processing, as in this case, you'll form a bigger band because it doesn't get rid of this red sequence, whereas in normal or canonical sequence of splicing, you will actually get rid of that. So you should see the presence of 2 bands if you in fact have knocked in, as we call it, a mutant SF3B1 into the gene. And that's just shown here. I'm not going to belabor these things, but you can see from the, again, mutants that Brian created in these various genes, which were predicted, because these have very predictable sequences of what they will add into the new transcript. You can see the presence of additional bands, whereas in the wild types and the controls, you just see the usual band that would be predicted. And unlike most things in our business, this may sound shocking to some of you, but it's really hard to reproduce other people's results. And so in cancer and biology, we actually were skeptical that, because people have shown this before, that you would do this if you overexpress this, et cetera. We could actually recapitulate every single known transcript, if you will, or RNA species that have been previously reported. What nobody had done prior to this was look at the protein expression. So it's one thing if you're looking at the RNA, remember DNA goes to RNA goes to protein. It's another thing to look at the protein, because the RNA changes would be expected to make new proteins and degrade other ones. And so that's exactly what Brian found. The blue and the red are the differential proteins that are being expressed between these isogenic cell line pairs. So who cares, right? It's great biology. We try to publish this in science, but what does this mean for cancer therapy? Well, again, whoops, I should have. We always want to think about how do these types of findings allow us to either diagnose, prevent, or treat breast cancer better. And this one has a number of potential implications in terms of therapy. So I think many probably in this audience have heard of Herceptin. I'm sorry, Herceptin. Have heard of Kadcyla, which is just Herceptin. It's an antibody against HER2 positive breast cancer, but it has a chemotherapy on the tail end of it. We call this antibody drug conjugates. There's a new one that just got FDA approved by a different company, where we have a different different chemotherapy on the tail end of the Herceptin molecule. I can't remember the trade name, but it's called DS8201. Thank you. I'm terrible generic name. At any rate, the premise of these things is that they attack things on the outside of the cancer cell. And so the idea here is if we're actually creating new RNA because it has this intervening sequence that didn't get taken out normally because of the mutant spliceosome, we can predict if that would lead to a protein that has on the outside of the cancer cell a new protein sequence. Why would this be important? Remember, antibodies have specificity. So if we can actually develop an antibody that could attack just this particular red protein, but not the normal form, we have now a targeted therapy. It would allow us to actually take cancers that have an SF3B1 mutation and express this particular protein and say, we can actually give this targeted therapy. So could that work? Perhaps. Brian actually looked first at the extracellular surface kind of catalog, if you will, of the RNA and the protein that we did in the analysis, and found that these were all supposedly on the outside. Again, you have to validate all these things. In particular, this gene and protein was present in a lot of different types of cancer cell types that he studied. So breast cancer, CLL, uveal melanoma, and regular melanoma. So we have now taken these sequences and we're working with drug companies to develop antibodies that would specifically recognize this one versus this one. We had some initial hits that were pretty exciting. Turned out they weren't reproducible, but this is still work that we're doing with collaborators in the industry. All right, so I should probably have backtracked. Brian had the clever turn. By the way, I always find scientists who love acronyms, and if you don't come up with a good acronym, nobody will take you seriously. So he called these MASAs for missplicing associated surface antigens. So MASAs are present in a very diverse mutant lineages, as we already showed. They're in pancreas cell lines that we've had naturally. These are not things that we modify. They're in melanoma cell lines, they're in leukemia cell lines, and they're also even in leukemia patients. So Brian had some patients who had these mutations. And again, we're just recapitulating physiologically, do these things really occur in cancer? So we're developing these antibodies. We're actually working with Asai and a number of other pharmaceutical companies to see if we can get antibodies specific to these cell surface extracellular antigens. The next one is actually strategy. It's a very easy one to try because it really just involves using already approved antibiotics. So the mutant cells, as I told you earlier, will actually create additional sequences within the RNA. Now, RNA has developed, or our cells have developed, this process that we call nonsense-mediated decay. What does that mean? Essentially, if you have what we call a premature truncation codon, something that would tell the cell that this is not the right protein being made. Our cells have evolved, this goes as early as bacteria in terms of the evolutionary tree, a mechanism called nonsense-mediated decay that will take that RNA and destroy it. And the reason behind that is because many of these proteins or many of these transcripts, if they were major proteins, would actually in theory be poison for the cell. So that's the concept of why our cells have evolved this particular kind of safety mechanism of NMD. There are proteins that we now know of that can allow, drugs that we now know that will allow these proteins to be made. Why would you want these? Well, it turns out that there are some diseases like cystic fibrosis. I think most people in the audience have heard of cystic fibrosis. One of the most common mutations is what we call a premature stop codon. And so if you give drugs, you can actually allow the protein to be made and actually kind of ameliorate some of the signs and symptoms of cystic fibrosis. Now, again, that comes at a price because you're also potentially turning on things that are poisonous to the cell. But in this particular case, this is what we want. We're going to take advantage of the fact that the cancer cell is making all of these aberrant RNA species that if they were made of proteins could in fact be deleterious to the cell. And so Brian rationalized that if we use aminoglycosides, which are antibiotic class, that this would do this. And in fact, that's exactly what happened. These are two aminoglycosides. This is the targeted wild type, and this is the mutant clone that we created. And these are 10-fold serial dilutions of antibiotics. And you can see these are just cells plated and then stained with a dye. And you can see pretty readily that in the cases of the antibiotics, you see a much more early killing, much less drug concentration if it's in the mutant cells. So this is something that we would see as a real easy adjunct to doing therapies against breast cancers that have these mutations. You could try just giving them aminoglycoside antibiotics. Now, these things are also kind of toxic to the kidneys and the ears, so we have to be a little bit careful. So it wouldn't be something we would try alone, but again, it gives us a combination approach in the future, if you will. The next strategy is trying to use gene therapy. Now, in cancer, gene therapy used to be this hugely exciting area back when I was a medical student, and then it fell from favor because it didn't work. And now people are starting to do this. We have some gene therapies for some diseases, even prostate cancer in a way. And so the problems have been, how do you get a gene therapy approach just into the cancer cells? And then how do you actually target just the cancer cells so that only the cancer cells will die? The approach we're thinking about here really wouldn't rely upon the fact that you have to get it just in the cancer cell. Just like a drug, you give the drug systemically, right? It goes through your whole body, and there's something different about the cancer cell that allows the drug to work where hopefully the rest of the cells, the normal cells, are spared. So the idea here would be is if we actually could exploit the fact that this mutation in the spliceosome gene will only make a protein if it's kind of in using this aberrant intronic sequence, could we actually preferentially introduce a poison protein into cells that have this mutant gene, but not in the wild type? So that's schematically shown here. We're doing this as a proof of principle where we're using a reporter gene, which we call green fluorescent protein. So if this is being made, the cells will fluoresce green. And the idea here is that you have a mutant SF3B1, the RNA species is allowed to be made and the protein's allowed to be made. But if you have wild type, meaning normal SF3B1, you'll get this premature stop codon, and that will undergo this NMD process that I talked about earlier. So all this is really doing is exploiting the differential splicing, the differential ways that RNA gets made if you have a mutant SF3B1. So we tried this experiment, and it was really cool that it worked. So these are the two mutant cell lines we made, and these are the isogenic controls, so the wild type. So they're exactly the same, except this one has, these two have a mutation in the spliceosome gene. And you can see under the microscope that these will express the GFP and these really don't. And so that now gives us a lot of leeway into how we're gonna think about exploiting this. Sorry about that. One is which we're gonna try to optimize this. There's still a little bit of background here. It turns out there are other probably gene sequences we could use that the mutant spliceosome will use preferentially. The other thing is that we're going to work on an inducible suicide gene. That would be the most easiest approach. But I also started thinking about how we already have antibody therapies for cancer. And what if you could express HER2? We know that from breast cancer, right? We have Herceptin and all these other drugs. Or CD19. This is on a lot of B-cell malignancies where we have a drug called blinatumomab. And so these are the things that we are now working on where we're going to put actual genes that are not usually made in these cells, have them being expressed only if they have the mutant spliceosome, and then we can give them already FDA-approved drugs to see if this will work. And then finally, the targeting new metabolic vulnerabilities. This has actually been published. When Brian looked at the proteome, as we call it, when you compare the two isogenic cells of the mutant versus the wild type SF3B1, one of the things he was struck by, there were a lot of metabolic genes that were up or down regulated. And so this was a very cool idea that he had that turned out to work. One of the genes that is involved in serine metabolism, so we all have different amino acids that are used to build proteins, and some amino acids are what we call essential and some are not essential. Non-essential ones mean that if we don't get that amino acid from our diet, then we can, in fact, our cells have alternative pathways that we can actually make that. So it turns out serine is a non-essential amino acid because of this enzyme. If you don't ingest serine in your diet, this enzyme PHGDH will actually take glucose and convert it into, as well as lactate, and convert it into serine. And so if that enzyme is broken down, and it turns out that's what happens in these SF3B1s, because they will actually aberrantly make RNA that cannot be made into protein. And so the protein level of this particular enzyme goes way down. Now, serine has become an essential amino acid just for these cancer cells. And so Brian put this to the test, and sure enough, you can see here, these are just what we call phase contrast. After a week in culture, if you deprive them of serine and glycine, there's a big difference between the normal cells versus the single mutant cells that Brian created. And that's been recapitulated in a couple different cell types that he actually did. And then finally, and this is probably the most exciting one because it actually has led to a clinical trial that is now just being started at Hopkins, and Vanderbilt's going to be part of it. One of the things that has been super exciting in the field of cancer, I think many of you know this, is immunotherapies. But what's also been a very disappointing and frustrating aspect of this is that we can't tell many times who's going to respond and who cannot. So my former boss, Bert Vogelstein, and others at Johns Hopkins had this seminal paper in the New England Journal of Medicine a number of years ago. Their hypothesis was when they looked back at some of the patients who had the best responses to immunotherapy, they have had what we now call a high tumor mutation burden. This actually, in retrospect, makes perfect sense. When you think about our immune systems, what has to happen is that the T cells, these are immune cells, have to recognize something that's foreign or an antigen on the tumor cell. And remember, the tumor cells come from us. So for the most part, they look like us. And we have certain things that we call checkpoints or handshakes that prevent the immune cells from attacking our own bodies. So there's about a dozen of these checkpoints, and the one that's been most therapeutically exploited is called PD-1 and PD-L1. Turns out if you block that with various drugs or antibodies, then this guy can recognize this guy, but it still requires something that is a tumor antigen. And again, most of the time, the tumor cell looks just like our normal cells. This is why we don't get autoimmune diseases for the most part, and patients who do have autoimmune diseases, there's something wrong with this handshake. So we have to be careful because there's a lot of side effects with taking off these handshakes. You can get real autoimmune diseases, you can get a lot of inflammation. But the whole point of here is how do we get something that is different in the cancer? How is this antigen recognized by the immune cell? So it turns out that it's kind of like playing the lottery. Remember, DNA makes RNA, makes protein. These are all proteins here. And so the more DNA mistakes you have, those are mistakes that the immune cells have never seen before because they're mistakes that happen just in the cancer cell. Not all of them are going to lead to a good immune response. But just like if you buy more tickets when you play the lottery, you have a higher chance of winning. So it turns out that there are certain types of cancer cells that have tons and tons of DNA mistakes in them, and that has led to this chance that you're going to form what we call a neoantigen. So this particular paper put on the map for the first time what we call tissue agnostic FDA indication. So any cancer type, doesn't matter if it's breast, colon, etc., that has a high mismatch repair deficiency type of mutation, meaning their cancer genomes have tons of DNA mistakes, can now get immunotherapies because the response rate to this is something like 70% or more, which is unheard of usually in our business. So our hypothesis was very similar. If you're making a lot of DNA mistakes, you make a lot of RNA mistakes, you make a lot of protein mistakes, and you respond to immunotherapy. In this particular case, we're showing that the mutations in this gene SF3B1 is making a lot of RNA mistakes. So even though the DNA doesn't look like it has tons of defect mistakes, the RNA certainly does, and the protein certainly does, as I just showed you. So we actually looked retroactively at 5 early trials of immune checkpoint immunotherapies. There were only 4 patients. Remember, this is very rare that have these hotspot mutations, but they all had a response, including one durable response. And so we've also seen at the Hopkins Molecular Tumor Board of lung cancer patients. Lung cancer patients can get these drugs no matter what these days, but this patient did not have what we call a high tumor mutation burden, but did have one of these mutations that had a dramatic response to immunotherapies. So this has led to Tasha Hunter, who is a fellow in my lab, is now at the University of Washington Fred Hutch Cancer Center as a junior faculty, doing what we call a virtual trial for breast cancer patients with these mutations. What do I mean by that? Well, as I showed you before, probably only about 1 to 3% of all breast cancer patients actually have a mutation in this gene. So trying to do this at a single institution or even multiple institutions would probably take forever to recruit these patients. Instead, what we're doing is setting up a web portal. We're going to recruit patients using social media, and if they have their genome sequenced, just have them fax it to us with permission, and then we'll take a look at it. And if they don't have any good therapeutic options other than trials, this is something that we're going to recommend to them. So this is really something that is the merger of 2 ongoing projects in the field. The first one being the metastatic breast cancer project out of Dana-Farber, led by Nikki Waddle's group. And for those of you who don't know this, this is a great example of what you can do with the internet. He actually rationalized that he wanted to just create a database, be able to retroactively get tissue samples and cheek swab samples from patients. And for metastatic breast cancer patients, once this got on Facebook and Twitter, you can see here, it just took off. And they had over 2,000 patients in 8 months. And Nikki's gone back now and actually collected tissues from all these patients, sequenced them, and now deposit them in the database. So this really shows you that we can, as a field, collectively recruit patients using social media and the internet. Similarly, ASCO has this, what I call the pragmatic version of something called the NCI MATCH trial. This is called the Targeted Agent Profiling Utilization Registry. A lot of pharmaceutical companies have donated their drugs to see whether or not patients who have mutations or genetic alterations in cancers, other than what the drug is FDA approved for. As an example, HER2 positive breast cancer, we do see HER2 amplification in some other types of cancers, but we don't really know, other than in gastric cancer, whether or not it works. So if you have HER2 amplification on a next-gen sequencing report of a patient's tumor that is, let's say, esophageal cancer, they can go on this trial and get Herceptin for free and see if it responds. So really, again, we're merging these 2 concepts. We're going to use an immune checkpoint inhibitor that's already been FDA approved, same one that was in the New England Journal paper. And this is the website. It's been created. It's not live quite yet. I think we got the final protocol approval just a couple of weeks ago. A colleague of mine who's still at Hopkins is actually now going to lead it there, and we're going to be a site at Vanderbilt. And of course, Fred Hunt with Tasha Hunter. So this is kind of what I've been doing now at Vanderbilt, where we're going to use and leverage the power of social media, telemedicine. We're also working hand in hand with genetic cancer specialists, because now more and more, even heritable types of gene mutations like BRCA1 and BRCA2 have therapeutic implications and consequences. And we're putting this all together, getting everyone tested, getting the data, convening our tumor boards, and then trying to actually get them into good trials or do off-label use therapies. And then in between, collecting all this data, eventually de-identifying and releasing this for public consumption, because everyone right now is kind of doing their own N of one experiment, as we say, but nobody's collecting and recording this data. And so it's my goal is that we'll all work together and amass a very, very pragmatic, useful data for clinicians out in the middle of nowhere, really, to say, hey, if I've got the internet, I can actually see if anyone's seen this before, they've gotten the drug, and what was the response. So in conclusions, hopefully I've shown you that MASAs, as we call them, represent potential targets for therapy or novel immunotherapy targets, that SF31 and other actual spliceosome genes, believe it or not just F31, can create targetable cellular vulnerabilities, that these mutant cancers may be susceptible to immunotherapies, that we can leverage what we currently have in social media and the internet to identify cancer patients who might never have otherwise even thought about getting genetic testing, and then finding these patients again the best drugs in clinical trials. And I think with that, I don't know if this is going to work. So I get a lot of criticism. We all do in this precision oncology field that, randomized controlled trials have shown this doesn't work, et cetera, et cetera. You can't do the traditional randomized controlled trials for patients with these because they're so rare and everyone's a little bit different. But I'm going to play this. It may not work. And if it doesn't, I'll show you the story, because this is how I view precision oncology right now. Oh, it's not. It is working. I had low hopes.
One day, a man was walking down the beach when he noticed a boy picking up starfish and throwing them into the ocean. Approaching the boy, he asked, "Excuse me, but what are you doing?" The boy replied, "Throwing starfish back into the ocean. The sun is rising and the tide is going out. If I don't throw them back, they'll die." The man laughed to himself and said, "But there's too many starfish on this beach. You can't possibly make a difference." After listening politely, the boy bent down, picked up another starfish, and threw it into the ocean. Then turning to the man, he said, "I made a difference to that one."
You have to forgive me for being so schmaltzy about it. I just love that story. That Bangladesh girl is that starfish. And that girl surely would be dead today if it not for the fact that we just identified her and got her luckily on that trial. And this is happening more and more. I have more than just that Bangladeshi girl as that story. And so with that, I just want to thank everyone who's been involved in part of this, several generations of lab people, collaborators at Hopkins as well as now at Vanderbilt University, and then a lot of different funding sources, including Susan G. Komen. Thank you for your time, and I'm happy to entertain any questions.
We're gonna go around. Yeah. Does anybody have any questions?
I have a question. I was wondering if you foresee a liquid biopsy to monitor for like recurrent breast cancer. Do you think that'll happen in our lifetime, where there'll be a liquid biopsy to monitor those mutations that can shed in the blood?
Yeah, I do. So the question is whether or not we can use liquid biopsies in something that we call cell-free DNA to actually know, in essence, who's cured and who's not. So this is a big problem in our business, and I think many of you know this, where we are over-treating patients, and we know that we are based on statistics, but we don't know who's cured and who's not. And so when we first started getting into this business, now probably well over 10 years ago, when I saw that it could work, that's the first thing that I started going after. So we have a trial that is accrued. It's through what we call the Translational Breast Cancer Research Consortium, which involved, I think, 16 or 17 sites through the TBCRC and then additional sites, 6 or 7 at Hopkins. 228 patients. We're looking just at triple-negative breast cancer patients that are HER2 positive in the neoadjuvant setting. The primary goal here would be if we can actually show that patients in their blood have cancer DNA molecules before neoadjuvant therapy, they get their chemotherapy, and if they clear it completely, does that predict for pathologic complete response at the time of surgery? So pathologic complete response means when the surgeon goes in, they don't see any cancer anymore in the lymph node and the breast. So I actually proposed this about 7 or 8 years ago, that if we could prove that, we could actually offer women in the future a no surgery option. I gotta tell you, the surgeons in that audience looked at me like, what? That's completely crazy. But it's not when you think about it, because therapeutically, we haven't done anything for them surgical wise, because we've done a lot from a prognostic standpoint, because we know that's a good prognostic indicator in those subgroups. But you just don't know who's achieved past CR. So that's the number one objective. The second objective, getting to your question, which I think is ultimately the much more important one, is we're following these patients out, serial blood draws, and it'll take time, just like any adjuvant therapy trial, to see whether or not the presence or absence of cell-free tumor DNA will prognosticate for relapse. That's something that people have shown kind of retroactively. When you look really carefully at the data, I question whether or not they're cherry picking their patients. But I think a prospective trial like ours, and there are many more that have been planned in parallel, will answer that. And I think it's going to be different or more challenging for different subgroups. As an example, triple-negative breast cancer patients we know usually are going to relapse within the first 5 to 7 years. And so we're focusing on that group first. HER2 positive disease and ER positive disease, we now know, usually tend to relapse if they're going to. Those patients are going to relapse kind of years to decades out. And so the challenge in our field right now is that we're trying to collect these samples serially, but we still have to follow these patients in a prospective fashion many, many years, if not decades, to prove that this will really work. I am hopeful by then the technology will prove that we can actually detect it more reliably and reproducibly. I think right now it's tough in these slower growing ER positive or HER2 negative tumors that have a long kind of latency period, if you will. Sorry, that was a long answer to your short question. So yes is my answer.
Dr. Toppmeyer, Dr. Blackwood, do you have anything extra to add to anything that Dr. Park just said over the last?
I don't think anyone could add to Dr. Park, but it was a phenomenal talk, and I really thank you guys for coming here. The liquid biopsy thing for us has always been something that we're interested in because it would be so nice. Patients, as you know, after they've been fully treated with surgery, chemo, and radiation, or surgery, AI, and radiation, or whatever combination you give to them, are always looking for us to monitor them. And as you know, I'm old enough, and maybe some of you are old enough as well, that we used to do CAT scans. And then, you know, obviously we don't do CAT scans anymore. Patients really want some sort of follow-up. The liquid biopsy story I've been following very closely because I do think there's something to this. I think there is the issue. I get that many of these studies have been stopped and started, and we can't prove it, and it's not ready for prime time. We've gone to many lectures on these, read many papers, but I do think there's something to this, particularly for follow-up for patients. I think there is something between recurrence to stage 4 disease, and there's something before that. And I think if we can catch those patients prior to true stage 4 disease, we will make a huge difference in their survival. It's just a matter of trying to figure out who and where.
And what.
Right.
That's the bigger question. Whether or not, you know, and this is why we don't need staging, earlier detection does not lead to better outcomes. But the question is, and if we understand what some of those targets potentially are, I think what opens up the door to do something like this.
I think our field has thankfully evolved our thinking where when I was first proposing these types of studies, there was such, I won't say backlash, but push for conservative approaches where patients, they said, this, we can't treat patients based on this positive finding. And I'm certainly worried about false negatives, that patients are going to recur despite having non-detectable cell-free tumor DNA. But at the same time, most of us in this business recognize now, especially with all the other data that's come out, that if a patient has real tumor DNA in their blood, notwithstanding some of the artifacts of what we clonal hematopoiesis, this is a bad thing. And so I'd always try to push a trial where if we find any type of tumor-associated cell-free DNA, we should give those patients something else. And we now have data with CreateX and other things where changing a therapy or adding a therapy may in fact change outcomes. That was also a big concern and criticism in the past. So Europe is kind of ahead of us. They seem to be a little bit more progressive in their thinking about, being a little bit more risky in terms of the trials. But I think the time is now to really start doing this because we have the information. It may not be perfect. I don't think it'll ever be perfect. But I think most of us would believe that if you have a tumor that had a certain mutation, and then after all standard care therapies, you still detect that in the blood, that's not a good thing.
Well, speaking of that, Henry Cure, as you know, has a trial now for hereditary amplified disease, and he's going to try and put us surgeons out of business with this trial. And what he's done is he's taken hereditary amplified disease, treated it with whatever chemotherapy approach plus anti-hereditary therapy. And then they repeat all their imaging and they repeat their biopsies. And if there's nothing left on imaging or on biopsy, they offer the patient an option not to do surgery. One of my patients actually came to me as a fifth opinion after she had been down there to possibly participate in this. I have to tell you, it's hard for patients to accept it. It's not just we, the surgeons, who are pushing it. But I think we're all getting to the point where we'd like to target everything and be more specific. I mean, the B51 trial is about post-mastectomy radiation and surgery. So, are we doing radiation? Are we doing any good to the patient anymore? And do they have residual disease somewhere in their body? And I think we've tried with PET CTs. I think we've tried with sort of other imaging modalities. And I do think it all boils down to this eventually, myself.
I'm biased, but I would agree.
We do have a question right here.
Explain a liquid biopsy. I don't know what that is.
Yes, so it's kind of a confusing term for sure. And liquid biopsy means different things to different people in the cancer research business. So the idea is that most of us think about taking a couple tubes of blood and then the blood component is spun out so that you just get plasma. So it turns out all of our cells shed or secrete free-floating naked DNA particles. They're just these small little fragments. Nobody knows really what they are. I actually think they're just waste products. I call it cellular poop. And it just goes into our bloodstream and in our circulation and goes out. So usually with cancer patients, even if you have heavily metastatic disease, the amount of tumor DNA in that, what we call cell-free space, the naked DNA floating around, is kind of the minority. Sometimes like 1 in 1,000, 1 in 10,000, maybe more. Because all of our cells, our normal cells, are also doing the same thing. So even though we've known about this for decades, we hadn't had the technology to really pluck out the proverbial needles from the haystack of cancer DNA from the normal DNA until now. And so this first got really put on the map by my former boss at Johns Hopkins, Bert Vogelstein, where he used technology that he created that allowed us to actually find cancer DNA from all the normal. And that eventually also led to this technology that we now all call next-gen sequencing. Other people might tell you, though, that liquid biopsy also includes something called circulating tumor cells. So these are actually cancer cells that can float around in the body, and we can pluck those and stain them for cancer markers like estrogen receptor and HER2, look at some of their DNA quality. So it's different things to different people. And liquid biopsies can also mean looking at cell-free DNA from the urine or the saliva or the cerebral spinal fluid. But for the most part, I think people, when I talk about it, most of the time we're thinking about blood, plasma, and looking at the tumor cell-free DNA fraction. And these are commercially available tests now. Whether we should use them is also somewhat controversial. We tend to use them only when we can't get a good tissue biopsy in metastatic patients. Yes.
How expensive is this type of treatment? Like some of these drugs, when they become mainstream, I was just wondering when you apply that, not only in this country where we have healthcare problems, but globally, what's the cost going to be to society? How are people going to be able to afford to have, this treatment?
Now, that's a great question. I could probably spend 2 hours talking about it, but the topic of financial toxicity has now been known. I think that's an unanswered question and a really difficult one to tackle. Drug companies have to recoup their R&D costs, and they will argue to you that if they don't do that, they're going to be put out of business. At the same time, if we have a drug like leterotrectinib that can have this amazing response, what is our ethical, moral obligation to treat a 14-year-old girl who's otherwise going to die if she can't afford that cost of $100,000 a year? I don't have the answers to that. My hope would be that part of what we can do in the liquid biopsy space, it all comes back to liquid biopsy, is give patients a dose or two. And then liquid biopsy is not only qualitative, looking for the actual mutations in cancer, it becomes quantitative. So if you actually see a level of cancer tumor DNA go from here to here, would that be good enough to say, for this patient, it's working? On the other hand, we spend a lot of money and healthcare dollars and time giving drugs that never work. And we wait 3 months for the next CAT scan to find out that it didn't work. And so could we actually, a week later, this is the trial we're doing right now at Vanderbilt soon, if it goes from here to here, meaning that the therapy was not effective after a week or two, could we actually stop that therapy and move on to something else? And would that afford a better outcome? So those are tough questions to answer, but we have to do that in the context of a clinical trial. But that, again, you could see if a really expensive drug that's not working for patient X, Y, or Z, we would stop after one dose and would really dramatically lower the cost.
And can I answer, PD-L1 in triple negative disease has become sort of the holy grail, like HER2 was in HER2 amplified. I've been to several conferences and talks, and people are talking about doing PD-L1 testing routinely on triple negatives. Where I assume that's not where we should be right now.
Well, I think it is in the metastatic setting because it is approved for first line, and you have to have PD-L1 positivity. It's a little bit weird, 1% in either the tumor cells or the stroma. And I think that's been the problem with these antibodies for the different cancer types. There's like at least 3 different antibodies, depending on the trial, what the drug company used, et cetera. There's not great correlation, certainly not perfect correlation about whether that predicts. And I think most of us in this business who have treated triple negatives with frontline immunotherapy and Abraxane have not seen the type of long-term responses that we've seen in other cancers like lung cancer and melanoma, some subsets. So we still have a long way to go. I don't know that what your experience has been on this, but I feel like there's still a huge room for improvement.
And I think, particularly in the neoadjuvant setting, given pembrolizumab, you can see a lot of toxicity, and you're dealing with a patient population that is potentially to do very well, and the long-term toxicity from these agents is significant.
Right.
So I think it's very different than a trastuzumab, where it is well tolerated, that the long-term toxicity, granted there's, the potential cardiac toxicity, but these agents are, they have a diffuse, broad toxicity profile of rare side effects, and these are not necessarily reversible. So I think we need to be, and as you know, in that neoadjuvant trial, PD-L1 it did not predict or response. So I think we need to have better biomarkers. I don't think that in breast cancer, not to be a Debbie Downer, but I don't think in breast cancer immunotherapy is actually going to be a home run in the way it has been in melanoma, et cetera. But, as your work speaks to, it's that one starfish, right? I mean, it's important that we continue, because, when we do find that one target, that may open the door to, as we, discussed, novel therapeutic interventions. And the example of lung cancer is one problem.
It is.
For someone who's a fellow, I don't want to say how many years ago, where we used drugs, and then we put patients on hospice. It's a brand new world.
Yeah.
So, and we're seeing that in breast
I
cancer
mean,
as well.
I tell fellows nowadays, if you ever told me that patients with metastatic lung cancer could live longer than a year when I was training, you would be just like, what are you smoking and can I have some? Because
Right.
that just was unheard of. And melanoma, metastatic melanoma, same thing.
Same thing.
Yeah.
So we do have a question right here and then another one right after.
I have a question about the biopsy. Is that gonna be a trendy, like liquid biopsy versus a traditional FNA or surgical biopsy? The reason I ask it is when we request, the traditional biopsy and the 90% will come back say, oh, the specimen is not insufficient, finding. So is liquid biopsy gonna be gradually replaced, the FNA or the surgical biopsy?
So my short answer, because I'm long-winded up here, is I believe so, but my long-winded answer is probably not for at least a decade. The reason for that is because current technology doesn't allow us to get to the same sensitivity as a tissue biopsy. And so when you look at these commercial liquid biopsies, our algorithm at Vanderbilt when I was at Hopkins is that if a patient can't get an adequate tissue sample, we can consider sending a liquid biopsy, with the caveat that if they don't have enough tumor burden, like actual tumor lesions, the likelihood of picking up some things are going to be lower and lower and lower. And the number of genes is only about 100 compared to 600 with the tissue biopsy. It really comes down to math. What I was saying earlier is that there's only a few cancer DNA molecules compared to normal in the blood, whereas in the tissue, you can get up to 100%, usually it's not that high. But with current technology, the time and cost needed to actually get enough DNA and sequence it down to the same level of sensitivity as a tissue, when we're comparing liquid to tissue, just isn't there yet. They could do it, but it wouldn't be pragmatic. You'd have to get probably, a pint of blood from the patient, and you'd probably have to wait weeks before you could, or more than weeks, probably several, a couple months or more to get all those sequencing done. It's just not pragmatic right now. But as technology gets better, faster, and cheaper, I think it will actually become like that.
Thank you for your wonderful talk.
Thank you.
I have a question, and it's kind of a dumb question for you, but maybe for us it won't be. We know that breast cancer is very heterogeneous, and when you have in your preclinical studies and clinical studies, you see a cascade of mutations. After you do targeted therapy, those mutations mutate. And what's that frequency, if you know? Because that's what I'm afraid of with that very targeted therapy that we do.
Yeah, you're absolutely right. And I think we don't have as many targeted therapies in breast cancer. Certainly we have examples like estrogen receptor, where we now know if you use aromatase inhibitors and maybe tamoxifen and some other drugs, you're much more likely, if you just use that as a single agent for years and years and decades, you're going to get estrogen receptor mutations. And so that allows the cancer cells to grow without estrogen because it's just autonomously signaling through the estrogen receptor pathways. Another example that people are now looking at, including our lab, but nobody I think has a strong handle on this yet, is the CDK4/6 inhibitor, so Ibrance being kind of the prototypical drug. There's probably multiple mechanisms for all of these things in terms of what are the genes that get altered and changed. But I do think most of us recognize that if you have a loss of function mutation, as we call it, so if you knock out a gene called RB1, that probably is a kind of resistance marker for all of these CDK4/6 inhibitors. And people have described that in case reports. We've certainly seen it in the lab, as many others have. So all of that is evolving, but I think you're exactly right. Every time we get a new drug in the metastatic setting, we're all thinking about what is the mechanism of resistance, because we're seeing progress for sure, but we're not seeing cures. And in fact, I'm proposing, and this will be another goddamn super crazy type of trial, the way we think about metastatic disease kind of is nonsensical, how we treat it. We give one drug, we just keep giving it till it stops working. And you think about it, that's a perfect setup for drug resistance. And what I've tried to propose is using liquid biopsies. Could we actually alternate therapies, even if it looks like it's working? Because if you hit it with different therapies at different time points, could you actually afford a better outcome in the long term? I mean, it makes perfect sense, but nobody's really thought about how to best do that. And so that's one of the things that we're trying to design right now, but it's challenging.
And when you're doing your virtual tumor board and you're do you actually, on a routine basis, get a genetic, like a foundation medicine report on your tumors, or is that only in that metastatic setting or triple negative?
So we don't generally for this breast tumor board, but I kind of stole this idea and I created a virtual international molecular tumor board. You all are more than welcome to participate. We have now, I think, 17 academic sites, I think probably 7 community hospitals and 5 international sites. And we also meet once a month. We use Zoom. I lead this, it's great fun. We get great cases, patients who have already had some sort of CLIA grade NGS. Turns out I have colleagues in Romania who they wrote a grant for foundation and they can get it for free now, but they don't know how to interpret it. And so this is a protege of mine when I was at Hopkins. And so we've been meeting at ASCO planning this for the past couple of years, and they routinely present their cases and we really dissect them out. We just had a great case from a colleague of mine at the University of Wisconsin-Madison, Mark Burkhardt, who showed us that a 60-year-old patient had secretory breast carcinoma, had the NTRK translocation that's now being lurbinectomy. And he went back to his pathologist and said, how come you guys didn't categorize this as secretory? And they actually apologized. They said, we're not pediatric pathologists. We weren't looking for this. And in retrospect, they now see the features of secretory carcinoma in that 60-year-old patient that has this translocation. Again, it's that starfish case, but it's she was on hospice. And so she's off hospice now. And I don't know what our next staging scan will show, but that's the type of stuff that we live for. We can help a lot of patients if we could just identify them. And using the internet and telehealth and telemedicine, which are kind of strengths of Vanderbilt, is really something that I want to push for the next 5 years.
So you're using a metastatic disease setting?
For the most part, it's metastatic, yeah. There are some indications, like in stage 3, whatever letter for lung and for melanoma now, where you could get BRAF in the adjuvant setting with BRAF inhibitors, that we're doing this. But for the most part, we're trying to be very judicious on how we use these. And it's nice that they put me in charge of like being the bad cop. Because if I see something that comes in that makes absolutely sense, it's not going to change that patient's therapy, particularly early stage. I think everyone knows now, don't try to order an early stage cancer patient because it's got to be approved by me. I think that's not the best use of resources. So, right now.
You have another question right in here.
Good evening. Thank you for your presentation. Within the communities of color where there's high incidence of breast cancer, how are you getting the word out for people to participate in the studies for protocols and to advance information gathering?
That's a great question. We just went through our big cancer center grant renewal, so we were getting these exact types of preparation for the questions. Another great thing about where we are at Vanderbilt is that the Tennessee and the whole Southeast, there's a lot of racial disparities in terms of healthcare outcome, particularly in breast cancer, and particularly in Black women who get breast cancer, especially at diagnosed at an early age. So there's an amazing resource called the Southern Community Cohort Study, led by colleagues of mine at Vanderbilt, where they collected a lot of information on the whole 8-state Southeast region, both their genomics, their genetics, their cancer samples, et cetera. And that's allowed us to really leverage that database to look at the genetics and the genomics. And I also have a colleague whose researches in healthcare disparities, Tua Powell, where she is also looking at not only the genetics and the genomics, but also issues that are modifiable, like lifestyle risk modification, like inability or ability to access the healthcare system. And what Tua is doing with this is an R1 funded grant that she has right now, is trying to develop what we call a prognostic risk score. You can almost think of this as oncotype, but more than just oncotype, using the genetics and the germline of the patients, and this is specifically young Black women newly diagnosed with breast cancer, look at all the other factors and come up with a risk score, something that's modifiable that we can then actually use prospectively to identify patients who are going to have a worse outcome, and then try to actually make interventions where we can. Obviously, we can't change the genetics of our cancer or who we are, but all the other things we might be able to influence. And then on top of that, Pam Hall, who's our Associate Director of Community Outreach, she has put together this phenomenal group of community advisors, where she actually looks for people of color, men, women on equal basis, and many of them are cancer survivors themselves, puts them all in various rooms together to help us design the next wave of clinical trials, including outreach. An example of this that recently happened was even though mammograms have gotten more penetration, including in southern Tennessee, lung screening for cancer has not, and yet that's an FDA indication now, CT screening for lung cancer if you have certain requirements. So someone came up, we had this kind of internal request for applications and ideas, and someone came up with this, what I think is a brilliant idea. We just put up these huge signs next to our breast centers that says, if you're getting your mammogram, if you're a smoker or a former smoker and you meet this eligibility, you can also get lung cancer screening by CT. It's covered. And so this has now sharply increased our rate of lung cancer screening. So I think that's the fun part about being at cancer centers, and you have great people who are thinking creatively. We know that there's a problem. Our health disparities in Tennessee is really awful right now, but I think we're all coming together to think creatively in how we can actually get more engagement both from the community and affect more people coming into our centers to get screened.
This has been wonderful. Dr. Toppmeyer, Dr. Blackwood, do you have any closing remarks? Anything else you'd like to add?
No, this has been amazing. I'm sorry that Sridhar is not here today.
Thank you.
Sridhar Ganesan is our Associate Director of Translational Research and also runs our molecular tumor board and
as a friend.
right, and Ben and Sridhar are good friends, and it was a fabulous talk. I think it really makes us so optimistic for the future, and you know where we have come, where we are right now, and where we're going. And I think it was a fabulous talk, and thanks for coming east again.
Thank you. Appreciate it.
And Ben, I thank you very much. I don't have any good bad jokes. I'm not a twin, and I don't know beer. But I am very thankful that you're here, and I really appreciate the level of the talk you gave. And I thank everybody from Komen for putting this together and organizing this, and it is a spectacular dinner even for gluten-free people like me. But I'm excited about this kind of stuff. I mean, this is what we went into this is for the changes that are happening. I mean, I would, in some weird ways, love for you to put breast surgeons out of business someday.
I think it's ironic that breast surgeons want to do less surgery, men want to do less chemotherapy. I mean, radiation want to do less radiation. We all want to de-escalate, and I think that because we've seen in our patients what some of our therapies can do.
Yeah, it's also what you're saying is what works and what doesn't work, right? And giving chemotherapy for somebody with an oncotype score of 14, even if they have 2 to 3 positive lymph nodes, isn't going to alter their outcome.
Right.
So
we can learn. That's one of the good things. Also, that social media aspect of this made me think about a couple things we can do here for our outreach to our patients, especially with our system of 16 different hospitals. So, thank you very much.
Thank you.
I'm Suzanne Corson. I'm the executive director of the Central Doctors, the affiliate of CDG Komen. Thank you all very, very much for coming. I hope you enjoyed this. Thank you so much, Dr. Blackwood, Dr. Toppmeyer, Dr. Park, for being here. And coming out in the rain, I have to tell you that I chose this before it started raining, but I think I might have picked my gift for the doctors, knowing that they would need an umbrella. So don't blame me. But thank you so much and have a great night. I know.